today we're doing respiratory system, which is particularly topical these days. We'll get to actually meet the actual cell that the coronavirus takes out and messes up your ability to breathe and all that. So by the end of today, if you haven't really thought much about like, why do people end up on ventilators, it'll make a lot more sense. And I am going to start though by going over our warm-up. So let's do that. Wow, no, that's not what I want here. Here we go. All right. I was pleased to figure out that I could put little animated GIFs in these things. So in the future, maybe there's going to be more little animated things. So let's go through. So this one, name a hormone, a tropic hormone produced by the pituitary gland. Yeah, there's a bunch. What are the possibilities here?
TSH.
TSH, thyroid stimulating hormones, definitely one. What's another one?
ACTH.
ACTH, which is talking to the adrenal gland. What's another one?
FSH.
FSH, follicle stimulating hormone, as well as luteinizing hormone. Together, they're collectively called the gonadotropins that are talking to the testes and the guy and the ovaries and the woman. So any of those would work. And then again, the gland control, TSH would be the thyroid gland and ACTH would be the adrenal glands. And particularly ACTH talks to the adrenal cortex. The adrenal medulla, the middle part, is the part that is activated by the sympathetic nervous system and releases adrenaline. The cortex, the outer part, is the one that releases, in particular in response to ACTH, cortisol, the stress hormone, glucocorticoids. All right, let's continue rolling down. What essential mineral is necessary for your body to produce thyroid hormone? So we talked about iodine. Iodine, you can't make thyroid hormone without iodine. It's why you have iodized salt now, right? Most salt you get in a little pack, it's like, it's got iodine added in. So nobody is gonna have iodine deficiencies. Typically if they're just sprinkling kind of the classic salt on your food. And then this gland, speaking of thyroid, this is the classic thyroid gland here. Remember I was saying it's the only gland that has like places that actually store the hormone outside of the cells. So when we look at this thing, I can annotate. You've got the cells that kind of make up the walls. These are like these T thyrocytes. But then you have these big hollow spaces, are called follicles, that are filled with this colloid that binds the hormone. So no other gland looks like this at all. You have pancreas, the islets of Langerhans are little clusters of cells amongst other cells that stain a little differently. The pituitary is going to have an area that looks like dense cells and then a back part that has this kind of clear part that looks kind of like nervous tissue. Ovaries and testes we'll look at in much more detail. You'll get to recognize those very quickly once we spend more time with them. So this one is pretty darn distinctive. Let us clear that. And I've got to stop that. Continue up. Name a hormone produced by the adrenal cortex. So what are some of the hormones produced by the adrenal cortex?
Cortisol.
So cortisol, which is going to basically, it's a stress hormone. It mobilizes energy resources, kind of shifts your body into this mode to deal with stressors. shuts down long-term maintenance and puts your energy into being able to react quickly and strongly to some stressor. Aldosterone, someone said, aldosterone is talking to the kidneys, telling the kidney tubules to pull back more salt back into your body. So I think that's it. So any questions about the warm-up?
A quick question, are aldosterone and ADH, are they different hormones or are those the same hormone?
No, they're very different. They often work together, but ADH opens up water channels and has the kidneys pull more water back into your body so you pee less. And that's independent of ADH. ADH tells the kidneys whether or not to pull back, conserve, or let go of sodium. And if you have both of them on together, you'll maximize your water retention, because not only are you pulling water back, but you're pulling salt back, and due to osmosis, you're going to increase the gradient that the water is going to want to come back. But they work independently. Again, although if you want to do maximum water conservation, you often have both of them being released. Like in the renin-angiotensin mechanism, you have both aldosterone and ADH released to kind of maximize keeping water in your body.
Cool, thank you.
Sure. All righty then. So I guess we'll dig into the respiratory system. Well, let me look. I had my list of things to cover. Let me see. Oh, my list here. While we're in Canvas, I added a homework. It's a classic homework here, the endocrine homework. So it's here under remote week 5, which is where we're at right now. And if you click on endocrine overview homework, it just basically says, oh, wait, I want to see the assignment. Dagnabbit, hold on. Here we go. It's basically list the major glands of the endocrine system. So the ones that are in your syllabus, the ones we were talking about in class. And then for each one, I just want you, it doesn't have to be like an essay. It could just be a list. But I want, what is the main hormone or hormones released by that gland? And then, in a nutshell, the function. I don't want like a big essay on the function, but what is the basic functionality of that hormone? What is it doing? Like TSH, stimulates, the thyroid gland to produce thyroid hormone or something like that. So sometime in the next couple of weeks, I think, when did I make it due? I made it due in 2 weeks, Monday and 2 weeks from now. It's not a rush, but in the next couple of weeks, I want you to sit down and just make sure that you formally go through each gland and think about what are the hormones and what are their basic functions. So that is the homework. And I haven't quite posted it up yet, but this morning I've been working on grade breakdowns and stuff. And I think I'm close. I'm close to figuring out how much the exams are worth, what percentage the homeworks are worth. Because right now the points are all kind of cattywampus. I've just been assigning points randomly because I don't know how this Canvas grading system works. Plus some of the points are in Pearson, which aren't even connected to Canvas. So in the next day or two, I will have that posted. And it's going to reflect pretty closely what we had at the beginning of the semester, something around, what is it? Somewhere around 80% are the major exams, and like 10% homeworks and 10% quizzes, something like that. But I will talk about that more. If you haven't done, there's a couple of people I saw, most people have done the Pearson assignments, those mastering A&P. I think they're really helpful for studying for the exams. That's part of the reason I've put the energy into them. But I am going to count them as homeworks. So you should finish those if you haven't, because those are also going to be factored into your overall grade. Not a huge amount, but some amount. Okie dokie. What else do I have on my to-do list here? Endocrine. Okie dokie. So respiratory system. Let's dig into it. So we always start with what is it doing for you? What is function of the respiratory system?
Transports oxygen and carbon dioxide.
So the transport itself is the cardiovascular system. The cardiovascular system is the thing that actually moves it from one place to another in your body. The respiratory system is doing something more like on the front end of all of that. So before you can transport the oxygen around, or you have to actually take the, so I'm getting the Sakai's yet. So you need to be able to get O2 that's in the air and get it into your blood. And that's going to require what we're going to call, let me do this, going to require what we call pulmonary ventilation. Which is basically taking the air that's in the room and bringing it into your lungs. And then what we call external respiration, which is basically O2 in the lungs is going to go into the blood. So we're going to talk quite a bit about pulmonary ventilation, about your lungs and how you manipulate the pressure so the air actually flows in and out of the lungs. External respiration, we'll talk a little about, not in too much detail because it's just an anatomy class. Then the transport, that is the job of the cardiovascular system. That once the oxygen is in the blood, then the cardiovascular system, little Benny blood drop does his thing. Once you get into the tissues, there's this thing called internal respiration. Here, basically O2, from the blood goes into the tissues. For our class,
1 p.m.
And again, in physiology, we actually spend more time going into the detail of how that happens, what kind of factors affect that process. But for you guys, it's just, we've already covered it. In the capillary beds, oxygen is going to diffuse across the walls of the capillary into the tissues so the cells can get oxygen. We're going to focus mainly on ventilation and then a little bit about, what's happening in the lungs in order to allow gases to easily move in and out of the blood. And let's now start with some of the basic anatomy. Actually, other functions. Actually, I should mention another function. What's another function? What am I doing now that requires my respiratory system and flows of air and bellows of my lungs?
Talking.
Talking. Vocalization. Right? In fact, if you can't breathe, like if you're choking and you're done, you can't breathe, you can't call for help. You're just sitting there turning blue, but you can't say, help me, I'm turning blue, I can't breathe. You can't actually talk without your respiratory system running. You know, that's the international sign of hold your neck, means I'm choking or I'm suffocating, because you can't actually call out and tell anybody that you're suffocating. So that's going to be part of the respiratory system. And again, this class, we're not going to go into it so much, or actually much at all, but pH balance is actually something else that the respiratory system does. By breathing deeper or more shallowly, you actually affect the amount of carbon dioxide in your body. And carbon dioxide actually turns into carbonic acid in your body. So by breathing more shallowly, you actually make yourself a little more acidic. By breathing more deeply and clearing out the stale air, you make yourself more alkaline. So the respiratory system is actually an active part of your pH balance system, just by adjusting the depth and the rate of your breathing, which adjusts the amount of carbon dioxide in your body, which actually affects your pH in a relatively dramatic way. You can go up or down by 0.2 in the pH just by changing the depth of your breathing. So now I think we're ready to go and start the anatomy. New page. Obviously, it's not going to be the respiratory system without the nose. The nose. And let's give him some lips. And then let's think about this kind of hemisected head. So we can think about what's actually going on inside. So here's the going in. There's this nasal cavity. Here's the tongue. Here's the oral cavity. Oh, I should have gone further back. A sec. Here's the tongue, oral cavity. This is my drawing here is a little crappy. There, let's put his chin right there. So things to think about here. You've got this region here. This is the nasal cavity. This part here, this division between the nasal cavity and the oral, this is the oral cavity in here. What is making up this division here between the two cavities?
Would it be the palatine bone?
Exactly. So that's your palate. That's your hard and soft palate. So and if you all remember, what's the anterior part of the hard palate is not the palatine bone. What was it?
Sorry.
It was the maxilla. Remember, first, the anterior 2/3 was the maxilla, the posterior 2/3 was the palatine. And then behind that, you get the soft palate, which we've introduced when we talked about the palatine tonsils. And then there's that little doohickey hanging off of the back of the soft palate. What do we call that little doohickey hanging off of the back of the soft palate?
Uvula?
The uvula. spell it better. Uvula. Right, so if I was kind of drawing this a little, here's somebody with their lips. And then you were looking, here's their soft palate, and then there's this little doohickey hanging off there. And then their tongue is down here. So this is the tongue. Uvula. And this is just supposed to be staring down into the inky blackness of their throat. Right, and we saw, remember, the palatine tonsils were kind of on either side of that, hanging out in there. There are different terms for different parts of the soft palate. You do not need to know them. You don't need to know the pharyngeal, there's palatopharyngeal arch and things like that. Don't worry about that detail. You should just know this is all like the soft palate. You know, that's that part that's floppy. Like if you want to pretend you're a cat, you have to make it vibrate. Like, I'm just kind of making my soft palate like flap up and down in the back of my throat there. So, and what's the official word for the lips? Anybody know what's in anatomy? What do we call lips?
Oris.
Oh, that's a good guess. Labia.
Oh.
So labia. And so like if I look at somebody, we call these the labia. Developmentally, you get this thing where the structures all kind of fuse together in the front of your face, and you get this weird little divot right above where the middle of your lips are. Everybody has, I'm sure, has seen that kind of that little divot right there. That actually has its own name. That little divot is called the philtrum. If you look at the nose, the little, like, the holes themselves, the holes where the air goes in, those are called the nares. These would be what I'd call the part of the hole that is accessible from the outside are called the external nares. You know, if you want to pick your nose, you got to stick your finger up the nares. The little structure that kind of flares out around that, they are called the alae, which just means wings. There's the little wings flapping up over your nostrils there. So, you know, and the nose has different names. People talk about like the dorsum, kind of the ridge on the back of the nose. The nose is supported by cartilage, which is why all of our skulls, you know, never had noses on them. After the person's dead, the cartilage is just more organic, soft tissue that decays and goes away. So all the structures supporting the main nose, supporting the alae, all that, it's all different kinds of cartilage. There's hyaline cartilage for the harder parts, elastic cartilage for the, you know, stretchy or kind of bendier parts of your nose. So you have kind of a combination of hyaline and elastic cartilage to support the actual structure of your nose. While we're here, if we look at how the labia, you want to make sure your lips, they flap up and down, but you want to make sure that they kind of stay put in general. So we have these little webbing things that actually anchor them. So let me show you that. So like if this is your oral cavity, you actually have this little webbing thing here. It's called your lingual frenulum. Like take your finger or your tongue and feel right underneath the middle top of your lip. You'll feel this kind of stringy thing there that is anchoring it. So it keeps your lip from flapping all the way up. Like, can people feel that? I don't see, I know that John was not doing it actually. Can you feel that little thing in there? That's your frenulum. You've also got one underneath your tongue so you don't swallow your tongue. There's a lingual frenulum. Oh, dag nabbit. Okay, hold on. I hate when I do this because then it's going to be on video forever and I'll be embarrassed. The one on your lips is the labial frenulum. The one on under your tongue is the lingual frenulum. So the lingual frenulum, you can feel that under your tongue. And it makes it so your tongue doesn't flap all the way back and you choke on it. So frenulums are important. So frenulum, just that little webby thing that keeps your lip from flapping all the way up and hitting you on the nose, or keeping your tongue from folding back and getting stuck in your throat. So that's important. So back here, we have these different regions now. We have, here's the nares, the external nares, leading into this nasal cavity, which is this area here. We have our oral cavity here. They connect in the back here. Kind of coming down to the bottom of the throat. Once we get down here, we are going to end up with the larynx, which is going to be the next part. So nasal cavity, and again, uvula, that little dealy bopper hanging there. The soft palate and the uvula are gonna reflect back up. Like when you're swallowing food, you don't want to get food up into your nasal cavity. So the uvula and the soft palate can fold up and block this entrance, and it will keep the food from going up into your nasal cavity, right? You obviously know they're connected in the back if you've ever like laughed when you're drinking milk or something and blow milk out your nose, right? So typically you're not blowing food out your nose because the uvula and the soft palate kind of fold up and block off the entrance to the nasal cavity when you are eating or swallowing, I should say. So right below the nasal cavity is the whole part we call the pharynx, which is kind of the fancy word for throat. And the pharynx has three parts. You should know the part that just has air, it's not where food ever goes to, is the oropharynx. The part that is right behind where your oral cavity is, where the food can come, this is called the, oh my God, I'm doing it again. This is what happens when you get up too early. The place where food comes is the oropharynx. The part where only air comes is the nasopharynx. And then once you get down to the lower part of the throat, it's called the laryngopharynx. So nasopharynx, it's the upper part where only air is. Oropharynx is where the mouth, the oral cavity enters in. Laryngopharynx is below that, right before you get to the larynx and the bifurcation that goes either down to the trachea, the windpipe, or esophagus down towards the stomach. We'll come back. Nasopharynx, we've met the nasopharynx. That's where the adenoids live. The pharyngeal tonsils were up in the nasopharynx. Nasopharynx is also where you find the entrance to the eustachian tube or the auditory tube. Remember that little tube that equalized pressure between the middle ear and the outside pressure? In lab today, we'll get to see, I actually found a nice little dissection that shows the entrance to the eustachian tube up here in the nasopharynx. And you can see actually where that little tube connects in the back of your throat there. In terms of the actual nasal cavity itself, we've kind of met it already. We talked about the conchi. I call them conchi. Some of the other things I was watching today was calling them conche. I don't know. Remember there was inferior nasal conchi that were their own bones or derived, supported by their own bones. What bone had the little full ridges to do the middle and superior conchi?
What?
Exactly, the ethmoid. And all of this is going to be covered with what kind of epithelium?
Simple, no, stratified squamous?
So no, if you kind of think about respiratory passages, so this isn't going to have to worry about friction up there. You know, you're not rubbing up inside your nose there. You've got much more worry about.
Pseudostratified squamous?
No, it was one of the ones I know you knew it at one point because it was a question on like your first lab exam.
The ciliated pseudostratified?
Yeah, it was a ciliated pseudostratified. Ciliated pseudostratified columnar. So remember, it's going to be so much faster if I just type it. Ciliated pseudostratified. I think I misspelled pseudo, but forget it. No, it doesn't look right. I remember, and it had what did it always have with it?
Goblet cell?
Goblet cells?
Goblet cells. So it's, again, pseudostratified because it looks stratified with nuclei at different levels, even though reality it's not. Let's put some goblet cells in here, which look kind of goopy. And cilia. And remember that the cilia are motile, meaning that they wiggle back and forth. And the goblet cells, as well as underlying mucus glands, are going to create a layer of sticky mucus. And these little cilia are always sweeping the mucus along. And they're basically always sweeping it towards the back of the throat. So if we can either look up in the nasal cavity here. This is like, you know, to your stomach. Here's to your lungs. Actually, it's confusing here. So I'm going to say, gosh darn it. So here are your lungs. Here's your nasal cavity. Here's your trachea. So you have pretty much any place that is only air that doesn't have food. It's going to be the nasopharynx in the nasal cavities. It will not be your oral cavity or your oropharynx or laryngopharynx, because those have both food and, I should add, here's your mouth. Goes in there. But then once you get past where you're just in the trachea, this is also covered with that respiratory epithelium. And so all this stuff I'm drawing in blue is that epithelium covered with that sticky mucus. So any dust that comes in has a good chance of getting stuck and trapped in that sticky mucus. And if it's on the upper parts here, it's getting swept downward to the back of your throat. If it's on the lower respiratory tract, it's getting swept upward to the back of the throat. Sometimes they call this like the mucus escalator, mucus elevator, because you just basically have mucus traveling up the trachea. So you've got this sticky trap for dust and crap and catching little things. Once they're stuck, they get swept to the back of the throat. And then every once in a while, you just swallow, and you're constantly doing, even if you're not thinking about it, you're constantly swallowing. Again, you probably just noticed yourself swallowing right now, because now that you're being conscious about it, it's like, oh my God, I just swallowed. So that's going to be a really important way that you are able to deal with dust in the air. There's lots of crap, lots of things floating in the air, dust, spores, whatever. So this respiratory epithelium is an important way of dealing with, you know, filtering out the dust from the air that you're breathing in. Part of the reason why people who smoke often have kind of nasty coughs is it paralyzes the little cilia. So if you paralyze the little cilia that are supposed to be sweeping the mucus up to the back of your throat, then the mucus just kind of builds up. And then the way to get rid of it is try to just manually kind of get it up out. So that is the job of this stuff. This is all the places in your nasal cavity, the nasopharynx, and then the trachea and bronchi, until you get to the very small. Once you get to the very small branches of the respiratory tree, then it just gets more cuboidal. But we'll talk more about that in a little bit. Back over here. I'm going to give you this word. It's not going to be on your test, but it's a word. This entrance where the oral cavity meets the pharynx here and gets into the oropharynx. The official word for that little gap at the back of the oral cavity is called the fauces. I think I was thinking about it just because every time I hear the name of that dude on TV now that who's talking about, you know, the federal coronavirus guy, he's like Dr. Fauci. So, but fauci is just like that little entrance from the oral cavity into the oropharynx there. Other things while we're still up here. Another way you can help protect yourself from dust is nose hairs, right? So the nares, those nostrils, have hairs that filter larger dust and lint and stuff. And those hairs, nose hairs, obviously need a fancy word if we're in anatomy. So these are called vibrissae. So vibrissae is the more pedantic way of saying, nose hairs. So again, that's another way. So we're going to talk about different ways your respiratory system has to protect itself from the dust. First line of defense is just these big nose hairs that are going to trap big pieces of lint or something. Then once things get up into here, stuck like fly papers in the mucus and swept to the back of the throat where you swallow it. I should also mention that mucus is antimicrobial as well. It actually has this stuff called lysozyme, which lyses the walls of bacteria. So not only is it sticky, it's also antimicrobial. So if a bacteria lands in there, it's got double protection. The turbinates or the conchae, if you remember, when you look into the nose, it's all crazy looking. And so that means the air is going to swirl all around. And that's going to kind of help ensure that dust hits the wall and gets stuck. Also, this really helps warm up the air. The lung tissue we're going to see is simple squamous epithelium, really the most delicate kind of tissue you can have. So you don't want to have kind of hot, dry air coming down directly into that tissue. So there's a lot of kind of conditioning of the air that happens up here when you breathe in through your nasal cavity. Conditions the air. Kind of helps warms, humidifies, filters. So by breathing into the nasal cavity, and there's actually a lot of blood vessels right near the surface to help warm the air. It's part of the reason why it's easy to get nosebleeds and why they don't like to stop, because you've actually got a lot of vessels right near the surface up in your nose there. So nasal cavity, conchae pharynx. All right, so that is the basic story up here. Now what we're going to do is continue south.
Before continuing south, a quick question just to clarify the ciliated pseudostratified epithelium. Where does it go exactly? Like, is it also in the oro and laryngeal cavities, or can you just clarify where it is?
Right, so it's respiratory epithelium, like oral. What kind of epithelium are we going to find in the oral cavity?
The stratified squamous?
Yeah, exactly. It's going to be stratified squamous. So in the oral cavity and in the oro and laryngeal pharynx, and then continuing down into the esophagus, that's all going to be stratified squamous.
Okay.
The places where you're going to find the respiratory epithelium is going to be in here, and then going down towards the lungs, the trachea down here. But the places where food can be as well is going to be stratified squamous.
Okay. All right, thank you.
Sure. All right. Ooh, he's evil, red eye. Let's see. Let's continue on. All right, so food can go in your mouth. So food comes down here. This tube goes two directions. It goes down to the stomach, which is the esophagus, and also goes down to the lungs, which is going to be the trachea. So we need to make sure when you swallow your food that it goes to the right tube. If you have the food going down the tube that goes down towards the lungs, that's called choking. So the way we get around that is there is in the larynx, which is kind of the beginning, the top of the trachea here, there's this little flappy do called the epiglottis. See, let me draw this bigger so I can, I'm realizing this looks pretty bad. So oral cavity, nasal cavity, soft palate, this is my larynx. Trachea to the lungs, esophagus to the stomach. And they're actually attached. They share a wall with each other. And there's at the very top, so this thing I'm drawing here, this is the larynx. The larynx, sometimes people call it your voice box, or because it's where you're going to find the vocal cords. The larynx also has the protrusion that gives you your Adam's apple, right? So if I tap on my Adam's apple, I'm messing with my voice because I'm shaking up my vocal cords while I'm talking. We're going to talk about the larynx in just a few moments in more detail. At the very top entrance to the larynx is this little flappy doodle called the epiglottis. Epiglottis, it folds down when you're swallowing and basically blocks off the entrance to the trachea. And when you're breathing, though, it stays open and lets the air go down into your trachea. When you swallow, one of the things that happens is the whole larynx rides up and it actually helps seal that flap of that epiglottis against that opening. So if you touch your Adam's apple and swallow, you can actually feel it ride up and then drop back down. You know, this, I have no idea. So I have people, can people feel it? I don't see if people are doing it or not.
Yes, I feel it.
You can feel it. So I want you to actually get a sense that you have this kind of active thing happening to make sure you don't choke. So let's look at the larynx in more detail now. The larynx is a pretty interesting structure. Again, because it has the epiglottis and also because it has your vocal cords. So one of the things sometimes people get confused about the spelling of larynx and pharynx. It's kind of a weird word. You know, it's pronounced like, you know, larynx. A lot of times people say like larynx or pharynx or something, but try to make sure you don't get that confused. It's not larynx, it's like larynx. Oh, so I'm drawing a bad version of it. So first it's got You know what? This is going to be so much easier with the, I'm going to use the visualization program. This is actually, I'm realizing this is something I have always had trouble drawing well because it's kind of tricky to draw, and it's going to be so much easier to just show you in the program. Hold on one second here. We are going to let's go into here. So what we're going to do is we just want to kind of be in the head and neck here. And we want to add in the respiratory system. Ka-ching. So this is the larynx right here. You can see it anchored right below the hyoid bone. So the thing that's kind of tan colored there, those are the cartilage that support the larynx. This big one right here that I just highlighted in green, in fact, I'll isolate. That one right there, that is called the thyroid cartilage. It looks, people say it's shield shaped. It looks kind of like a shield. You know, you could carry it into battle and deflect arrows as you fight the good fight. So this is the actual, what we would call your Adam's apple. It's the thing that is sticking out in front of your neck. This is your Adam's apple. Below it is another one. It's called the cricoid cartilage. Cricoid, you can see it up in the corner here, cricoid, C-R-I-C-O-I-D. And the cricoid cartilage actually goes all the way around. It's like a ring. So like if I isolate this one, you can see that it's actually a complete ring that's sitting underneath the thyroid cartilage. So those are like the two main, the big daddy rabbit cartilages that make up the larynx. This space here between the two, this is actually one of the places, I can use my, where's my annotator? Oh, actually, I could just use my tools here. Like this space right in here. This is if you're going to give somebody, I think they call them Crike tubes. If somebody can't breathe, you know, you've heard of like a tracheotomy where they actually, you can cut a hole in the trachea to try to restore breathing. But a place they often stick the tube is actually right here in between the cricoid and the thyroid cartilage here. You don't want to, even though people do it on TV all the time and use a pen to save somebody's life who's choking on an airplane, supposedly it's trickier than it looks. Like I've had people in class who've done it and know how to do it, but they were saying like, you shouldn't just go ahead and try it because you've seen it on TV. So, I'm gonna get rid of those pens there. So that is those two main cartilage. And again, you see it hanging off here below the hyoid bone. Ooh, I want to get rid of those pens. How do I get rid of it? Oh. All right. I'm still learning how to, clear the pen. One of the things I recommend, if you haven't, go through the tutorials. There are so many. There's like 50 tutorials, but they're all good. Like you can draw on the picture 2D, in which case then when you stop your pen, the thing stays there, but you can actually draw in 3D. You can go to 3D drawing, and then you can draw on this, like, it's like, let's say I want to like, why is this not working now? Oh, it's because I'm not sure what's going on now. Whatever. All right, it's for whatever reason it's not working. I had it where you could actually draw and then rotate things around. It was really kind of fun. You could draw on the actual bones and stuff. So let me clear this. I'm getting trapped and turn this off. All right, so if we now look, let's get rid of this skeleton. What you're looking at here now is just the whole, you fucker. Oh, sorry. You're looking at the throat. I'm going to get rid of the throat. This is the pharynx, the oropharynx here. We're going to hide. Let's get rid of all this stuff. What you're seeing is I'm getting rid of, here's the laryngopharynx. I'm going to hide that. Get rid of that little doohickey. Now what we're starting to see is the epiglottis. So that, what the heck? This is funny. I actually spent time learning all how to navigate, and obviously I still don't totally know how to navigate. Dagnabbit. I'm having trouble rotating it, and I do not know why. So let's just isolate that. So the epiglottis, let's get rid of that bone. So the oh my God. I'm gonna just start a new tab. Let's just start from scratch. I do not know what the heck I just did here. This is frustrating. I'm going to draw a dagnabbit. I apologize. It's really pretty to look at it in there, but for whatever reason, I am being a total spaz right now on there. I totally recommend you go in and look at it, because what you'll see is there's the little epiglottis, which is sitting kind of right above. There's going to be the thyroid cartilage. There's the cricoid cartilage, which goes all the way around. And then the epiglottis is supported by elastic cartilage. The epiglottis is actually, when you go and you find those slides of elastic cartilage in our slide drawer, it's actually the slice of the epiglottis of some little animal. So the epiglottis is just this little cartilage that supports that thing and can flap back and forth. I am now going to look inside. Listen, if we do a cross section of the larynx, this is like looking down into the larynx. So again, like if this was the larynx with the epiglottis and the trachea below it, and here's coming down from your throat, what we're looking at here is like looking down into the trachea. And what you're going to see they're kind of cutting across, they're kind of around here. These are called the vocal folds or vocal cords. The little space in between the two The space is called, this is the glottis. And you have some more cartilages that actually anchor the vocal cords called the arytenoid cartilages. And they actually can move. So you can swing the vocal cords back and forth in order to open or close the glottis, which is really nice because then you can actually block it. Like if you're trying to build up pressure to cough, you can close the glottis and then open it up and let the cough out. Or you're trying to bear down for a bowel movement or whatever. You can shut that and you can build up pressure there. Other things about the vocal folds is they can change how much tension is on them. So they'll do faster or slower vibration depending on the tension. And the tension is also something you can change by doing muscles that actually kind of rock the whole larynx back and forth. So when you have your voice, when you're talking, the vocal cords, all they're really doing is vibrating and making kind of a buzz. You can change the frequency. You can go just by changing how tight they are, how much tension is on them. But all the rest of your talking is all the additional reverberation just in your oral cavity and the f's and p and ooh, ah, ah. That's all just changing the shape of the resonating chamber inside your mouth and your face. Right. Obviously, if you have a cold and the sinuses are clogged up, you sound very different too, because that's changing the resonating chambers. That all your vocal cords do is vibrate and make a buzz, and you can control the pitch. Right above the vocal cords, there's another kind of ridge that you can see that don't actually do anything. They're called the vestibular folds. So we've got that going for us. So vestibular folds. Let's see. And is there anything else I want to say about the larynx? The larynx has that where the thyroid cartilage goes out into the front of your throat here. That they call the laryngeal prominence. So again, what is a more common name for that?
Adam's apple.
The Adam's apple. And as part of secondary sexual characteristics, for guys, the larynx enlarges quite a bit compared to a female. And that causes two things. One, it means there's a more protruding, prominent Adam's apple or laryngeal prominence. It also means that the vocal cords end up also getting kind of larger and kind of thicker, and that's going to cause them to vibrate at a lower frequency. And that's why guys tend to have lower, why we have lower voices, it's just because of the physical properties of the vocal folds post puberty. They become kind of thicker and heavier, and they vibrate at a slower frequency. So that's like that changing voice. I'm going to try one more time to see if I can make this work, because, I'm going to go share the other screen one more time, because it really is way better than I can draw it. Let's turn off the skeleton again. So again, here, this whole thing, this is supposed to be the entrance into the pharynx. This is the oropharynx. Here's the uvula hanging down. This would be like the fauces. Oropharynx, laryngopharynx is below. And now I'm going to get rid of the structures that I don't want. Okay, and now you can see the epiglottis sitting there like a little flap, and how it's going to be able to flap down over the, oh, let's get rid of the nose too. Hold on. We'll come back. Actually, let's look at the nose real briefly. Here's all the nasal cavities. What are these things? What do you think these little tubes coming off of the nasopharynx are here?
Is that for the ears?
Exactly. Those are the eustachian tubes there. Those are the eustachian tubes. Over here, what do you think these are? Coming into the nasal cavity here. Here's the nose.
Is that from the eyes?
Exactly. Right? That is the lacrimal puncta going into the nasolacrimal duct, going down into the nasal cavity there. So this is the drainage of the tears. That was, I think, one of the questions, like, why do you have to give somebody a tissue when they cry? Because they're getting lots of tears all draining down here into the nasal cavity, and then you're going to blow it out the nose. Okay, let's get rid of the skull. Now let's get rid of all doofus. This, for whatever reason, my navigation, what? Let's just hide that and hide that. I want to be able to look. Oh, I'm gonna have to just eat. Oh, multi-selection, hide. Oh, so there it is, select area. Okay, now I can look down. There we go. There you can see these arytenoid cartilages. It took a while. All right, let's, so here's the epiglottis, the cartilage that supports the epiglottis that can float, you fucker, excuse me. That can float back, flat back and forth to block the entrance. These are arytenoid cartilages, which are, these little cords here. These are what the vocal cords are anchored on here, right here. Where are they? Click. So when these go back and forth, you can change the glottis. So you can see that would be the glottis, is that kind of little space in between. That's the entrance down. So this actually is really nice to get a sense of how the cartilages connect up. And normally we have a few really nice models in lab that you can play with and that are three-dimensional. I mean, the reason why I am belaboring this in the app right now is because I want you to go in and rotate it around and really see how it looks, because it's actually really cool once you get a sense of the moving parts. Epiglottis coming down from the top to block off the trachea when you're swallowing. The arytenoid cartilages swinging in and out to swing the vocal cords in and out to open and close that slit between the vocal cords. There's two more. There's a little corniculate cartilages. Don't worry about them. I could ask you about arytenoid cartilages on an exam, because they're pretty cool. They're the ones that change the glottis by anchoring on the vocal cords. You should definitely know the thyroid cartilage, the main big cartilage, that's your Adam's apple. You should definitely know the cricoid cartilage, the ring-like one that's all around the bottom. So now I feel okay going to the trachea. So, and you can see the trachea is going to branch into the bronchi into secondary bronchi, into tertiary bronchi. It's going to be like 23 levels of branching. So let's do that now. All right. That was more painful than I was hoping, but hopefully, and thank you for being patient here. So below the larynx, we get to the trachea, which people often think of just as like the windpipe. And we have to really worry about keeping your airways patent. Patent means keeping them open, making sure there's airflow. And in order for that to happen, because air doesn't have the same kind of pressure pushing out like that blood would have, you don't need to worry about an artery staying open because the blood is pushing against the walls, keeping it open. Or even your digestive tract, there's food's got some substance and you're squishing it along. But for the respiratory passages, there's got to be some reinforcement to keep the walls open so the tube doesn't collapse. And again, this word, keeping your airways patent, means keeping them open so the air can flow. And so we've seen it. It was our first introduction to hyaline cartilage in the beginning of the semester. In the submucosa of the trachea, there is cartilage, these cartilage rings. So if we looked at a cross section of this, there's cartilage. And they're not actually rings. They're more like C-shaped. You don't want them to be all the way around because you've got the esophagus right behind. So as the esophagus has a big ball of food in there, you wouldn't want to go whack, whack, whack on the esophagus is going right down the back here. So it needs to have the ability to kind of expand a little bit into that front part where the trachea is there. So trachea, and then it just starts branching and branching and branching and branching. So let's talk about the branching. The branches are called bronchi. We're going to have primary bronchi. These are the ones that go to the right or the left.
2 p.m.
By the time we get to this point, because there is no esophagus behind anymore, you can just have complete cartilage rings going all the way around. Basically, hyaline cartilage supports it. And then we start branching secondary bronchi. And these are called lobar bronchi. These are going to enter the main lobes of the lung. And you have a different number of lobes on the right and the left. Why do you think you have 3 lobes on the right, but only 2 lobes on the left?
Because of your heart?
Your heart?
Exactly. The heart takes up space on the left, so there's not as much room for lungs. And then actually this spot right where this branch happens here, it has a name. It's called the carina. Supposedly it has a lot of receptors on there. Like if something touches the carina, you'll go into like violent coughing, which makes sense. It means food has just fallen down your your windpipe and hits here, and you're going to try really hard to get it out so you can, your airways aren't clogged up with food. These keep branching again and again and again and again. The third ones are called segmental bronchi. Segmental bronchi because they go into what are called bronchopulmonary segments. The bronchopulmonary segments are clinically important because they're walled off with connective tissue from each other. So when you have some infection in lungs, often it'll be restricted like to a bronchopulmonary segment. So it becomes clinically relevant knowing that these exist. And then it just keeps branching more and more and more. There are 23 orders of branching. So into tinier and tinier and tinier and tinier little divisions here. All of this traveling, we call this the conducting zone. None of this is gas exchange yet. This is just bringing the air down to where gas exchange will occur in the alveoli. So all of this branching, as we get into the smaller, so let's assume we're getting near the end here. The small branches are no longer bronchioles. They are now, no longer bronchi. They're now bronchioles. And bronchioles no longer have cartilage in their walls. They just have smooth muscle is enough to kind of keep them open. They don't have respiratory epithelium anymore. Usually it's just some kind of cuboidal epithelium. So like, see if like smooth muscle. And then the lining is kind of cuboidal epithelium. This smooth muscle, can be dilated or constricted. Sympathetic activation is going to do what to the bronchioles? Is it going to make them get wider or make them more narrow?
Wider.
Yeah, wider. So it's going to make it easier to bring more air down so you can get more oxygen to supply your muscles and sprint away from the tiger. Parasympathetic will constrict the bronchioles. Like if you inhale some noxious smoke, it'll constrict these and try to limit the amount of noxious stuff that's making it down into the lungs. The final stop, we call the terminal bronchi. Terminal just means kind of end of the line. And that's going to bring us to the alveolar sac. So these individual little grapes are called alveoli. This is one alveolus. And then many alveoli get clustered together in these things called alveolar sacs. And there's different terminology, like the little hallway in the middle of it, they call an alveolar duct. But this is where we're going to call the respiratory zone. This is where, oxygen enters the blood. And CO2 leaves the blood, for that matter. So it's, I got kind of a little behind here. It's 2:06. let's say at, let's do 240. We'll come back and get into the alveolar sacs. Look at what's happening there. Again, that's where the coronavirus is going to strike. We'll see what it does to some of the population of cells, the little surfactant cells in the alveolar sacs there. And then we'll talk about the overall like lungs and pulmonary ventilation with the pleura in your thoracic cavity. And then we'll dig into the lab stuff. All right, so just briefly review what we've been looking at. This is just showing in a very pretty rendition the primary bronchi coming down, branching into the lobar bronchi, branching into, again, it just branches and branches and branches and branches and branches into the lungs. We're going to look at lungs in more detail in a little bit when we get to the lab. But yeah, when you look at an actual lung, if you cut the kind of indentation of the lung where all the stuff kind of comes in and out is called the hilus, like H-I-L-U-S. That's a generic term. Any organ that has a place where things kind of enter and leave is the hilus. Actually, lymph nodes, we call it, they have a hilus. Or the kidneys have a hilus. Hilus is just kind of that place where the stuff is coming in and out. We'll look at this in more detail later. But basically, you're going to have place for the bronchi to come in. There'll be thickest walls with the cartilage. There'll be the arteries, the veins. There's going to be two veins. Remember, two pulmonary veins coming out on each side, coming into the left, back to the left atrium. And this a bit prettier picture of what I've been drawing all along. The nasal passage, nasal cavity, down into the nasopharynx, oral cavity into the oropharynx, laryngopharynx. Here's the epiglottis, kind of protecting the entrance to the larynx and the trachea. We're going to look at this in more detail, but this is implying the lungs have this membrane around. They have the pleura, the serous membrane, which is going to be very important soon. The fact that the lungs have this two-layer thing, just like the heart had. The pericardium was a two-layer as well. This is a very weird picture, but it's showing like if you could see under this dude's skin, you'd see his nose is actually made out of cartilage. Again, this is showing those conchi, the lower, the inferior, the middle, the superior. You can even see the vibrissae, the nose hairs into this dude. You can see the hard palate and the soft palate that are separating the oral cavity from the nasal cavity. You can see the little uvula hanging off the back. You can see how it can just reflect back. And so when he's swallowing food, it's gonna block the food from going up so he doesn't sneeze it out his nose. It's also, yeah, remember, in fact, right here, this, let me get a better, maybe a darker one. Right in here, this is the entrance to the auditory tube, the eustachian tube. That's how the middle ear connects with the back of the throat to equalize the pressure. There's a little hole right there. I didn't really talk about this just now, but we've mentioned it in the past when we did the senses. What do we find right on the roof of the nasal cavity here?
Crista Gali.
Say it again, Elsa.
Crista Gali.
The crista galli is on the other side. That's the ethmoid bone is making the roof there, but the crista galli is not what I'm looking for here.
Sinus?
No. That's where you find the olfactory epithelium. That's where you have those special olfactory cells. They're right along the roof here. And then they have their little axons going up through the cribriform plate of the ethmoid to synapse in the olfactory bulbs there. So right on the roof of the nasal cavity is where you have those olfactory cells for the special sense of smell. So yeah, they'd be like right up here. And then this is actually showing like the little holes in the cribriform plate of the ethmoid to allow those axons to synapse. This is showing tonsils, right? Pharyngeal tonsil, palatine tonsil, lingual tonsil, just so you can orient. But then you can also now see here's the epiglottis. This lower one here is supposed to be the vocal fold. This upper ridge is supposed to be the vestibular fold. What is this, do you think? This kind of globby tissue right below the larynx here.
The thyroid?
Exactly, exactly. That's the thyroid gland. Yeah, this is like if somebody took a machete and just sliced your face off. It's so spooky. But this is showing those conchi and how when you breathe in, the air gets all swirled around, and that's going to help make sure the dust hits that mucus covered epithelium and trap the dust. And these hollow spaces, these are sinuses. These are like the maxillary sinuses here. There's ethmoid sinuses. Those are actually also, when we look at the nasal cavity, those all have little connecting tubes. Remember I talked about sinuses in the sphenoid, in the ethmoid, in the maxilla, in the frontal bone. These mucous membrane lined hollow spaces all have little connecting, I don't know, tubes, little connecting passageways to equalize pressure with the nasal cavity as well. When those little passageways get blocked due to inflammation or something, that's when you can get pressure buildup in the sinus headaches and stuff. It's like, let's move on. This is again showing the larynx, and here you can, yeah. Now I do think once you've seen it more in 3D, then it makes sense. The thyroid cartilage, more like the shield in front, this part. The cricoid cartilage, which is the one that goes all the way around underneath. This is the elastic cartilage that supports the epiglottis, the flappy dude that can come down and keep the food from going down your windpipe. Again, the thyroid cartilage is what protrudes to become your laryngeal prominence or your Adam's apple. And again, it's hanging kind of off of the bottom of the hyoid bone here. So this looks like he's wearing like one of those old hats from the Revolutionary War or something. This is showing how the arytenoid cartilages can swing back and forth and therefore close or open the glottis, right? This is like if you're trying to take a poop and you're building all this pressure, you don't want the air out, you have to shut the glottis. When you want to open and let the air flow freely, you swing your arytenoid cartilages and open the glottis. This is showing in that stratified, pseudo-stratified columnar ciliated epithelium. And this is also showing how the cartilage is supporting this with the trachea in front. This would be anterior, and with the esophagus in the back would be kind of posterior here. I mean, this is something we're not going to do. It's actually really fun. Usually with our little fetal pigs, we go in and we like snip out a bit of trachea and esophagus about the size of a piece of macaroni. And you can really see the difference. You can see the esophagus is this muscular floppy tube, and the trachea is held open, and it just stays patent, stays open because of the cartilage in there. And this should look super duper familiar. We saw this slide many times in the first weeks of class. There's the classic respiratory epithelium, pseudostratified columnar with cilia. Here, what kind of tissue is down here?
Hyaline cartilage?
That's hyaline cartilage. You know, a classic, when we look at the esophagus, we'll see it's very different. It's a tube, but it's gonna have very different kind of epithelium. It'll have stratified squamous. They'll have very different stuff in the submucosa. There's not gonna be any cartilage. So, you know, that's gonna, you know, classic kind of question. Like some of the slides we have are like, you know, esophagus and trachea, and you should be able to look at this and know immediately this is so trachea and not esophagus. So definitely you want to look at this and be able to make sure you realize why all the clues are telling you you're in the trachea, the epithelium, the cartilage. This here, these are just kind of, this you couldn't tell for sure. These are basically mucus glands that are underneath in here. This is just showing the little air passages as you get into the lungs. This is actually lung tissue. We'll look at this in more detail in a little later when we do more of the lab part. But this is where we're gonna take our discussion now into the alveolar sacs. So, you know, alveolar sac is, you know, these are like grapes. People talk about them being like a cluster of grapes, and each little alveolus is like a little balloon, and this is kind of showing them there. We're going to look in particular at one of them, and they obviously have to have capillaries all around, because that's how the gas exchange is going to happen. The whole point here is oxygen leaves the alveolus into the blood, and carbon dioxide leaves the blood into the alveolus for exhalation. This is showing the little blood capillaries right at the little corners here. And these are our little alveolar macrophages that are going to be the death cells. And these are the septal cells. These are the ones that the COVID gets. And we're going to look at those in more detail in a few moments. Okay, and we'll come back to that. Boop, boop, boop. So alveoli. You got a lot of them. There's something like 300 million alveoli in the lungs. So you get a huge surface area. The surface area is somewhere on the, I think they say like a small racquetball, like a racquetball court. Actually, I think I actually have the actual 750 square feet. I wish I had it in meters here. This is old notes, obviously. So you got a huge surface area for gas to cross and to absorb into the blood. And you know, that's why you have such a crazy amount of branching. And So what kind of tissue makes up the capillaries that are coming in?
Simple squamous.
Exactly. So here's my capillary. You know, the blood is blue as it kind of comes in. It's going to turn red as it comes out. It's got simple squamous epithelium. The wall of the alveoli are also simple squamous epithelium. And not only that, this is what's kind of cool here. I don't know if you remember, a epithelial tissue always has to sit on a basement membrane. So you have like the cells, and then there's this basement membrane, which had a reticular lamina and a basal lamina that were partly generated by the cells, partly generated by the underlying connective tissue. But there was this basement membrane. It turns out that these two epithelium share the same basement membrane. So you've got the cells from the alveolus, and then you have the basement membrane for that, but then you flip it on its head, and that's the basement membrane also for the the simple squamous cells of the capillary. Maybe I should draw those red to make this picture make more sense here. So there's a shared basement membrane between those two simple squamous epitheliums. So you have basically the thinnest possible distance possible to make using the things that we have to build humans. So we minimize the distance that the oxygen has to diffuse to get from inside the alveolus to the blood cell where it's going to pick up the oxygen and deliver it. Now, another cell in here are alveolar macrophages. Let's put those in, because then maybe I'll just type it so it's easier. AKA, and I love the dust cell. Patrolling the wild ranges of your lung tissue, looking for dust, any kind of particulate matter that might have gotten down in there. These are basically these big eaters, big white blood cells who are, you know, their patrol is the lung tissue, and they capture stuff and they can take it out to lymph nodes and stuff like that. So the last thing we need to add in here are the surfactant cells. So maybe I'll make them blue. They tend to be more cuboidal. And to understand surfactant cells, we need to talk a little more about water tension. Or surface tension of water is what I should say. So kind of spoiler alert for when you're taking chemistry, if you haven't already taken chemistry. Water, everyone knows water is H2O. And it turns out that there's kind of a charge imbalance. The electrons, which are more negative, spend more time near the oxygen. So the oxygen side tends to be more negative. The hydrogen side tends to be more positive. And opposites attract. So I've got some other water molecule. Here's an H2O, and here's an H2O, and here's an H2O. And for every H that's more plus, whoops, it's going to end up getting attracted to the oxygen that's more negative on some other one. So all the water molecules are all holding on to each other, pulling on each other. We call it hydrogen bonding. So all of these water molecules are experiencing these attractions. It's why water stays in a drop, right? That's why the water molecules don't just go do their own thing. They're all pulling on each other. Particularly if you just have like a surface of water, you have all the water down here pulling on each other, but on the surface, there's nothing to pull up on top with to distract them. So they're all pulling on each other really tight right at the surface. So you actually have the water molecules forming this kind of tight sheet at the surface of the water because they're all attracting to each other. Have people seen examples of this? I can demonstrate it if it's useful. How much do people have a kind of intuitive feel for this, or would it be useful to see how strong this really is?
I feel good on the concept.
Anybody else? Hello?
Yeah, I've seen it.
Okay, so yeah, I mean, especially if you've seen like a little water skeeter walking on the water, the reason they can float, or if you float a needle on a glass of water. So the water's pulling really tight. So we go back to our alveoli. The water in here is all pulling on itself, and the overall effect is it's gonna collapse in the alveoli. So just due to the attraction of water to itself, it would actually be impossible to inflate your lungs because it's the water, especially at this scale, those forces are enough that you can't fight them. And surfactant, the stuff that is made by these cells, actually goes and disrupts the attraction between the different water molecules. Basically, surfactant, like if I had my H2O here, that has like kind of the more charged and more charged areas. Surfactant kind of has its own charge, but then it has kind of a neutral thing. So the surfactant basically goes on and it interacts with the water, but then the other side of it is just this kind of neutral thing that doesn't attract anything. So it basically, this would be like my surfactant here. The surfactant molecules are basically going to isolate each of the waters from each other so they don't have attraction to each other anymore. So it breaks all of those attractions. So now all of a sudden, these aren't pulling on each other, and now you can inflate your lungs. So the surfactant cells are going to make surfactant, which breaks the surface tension of the water, which I can type. So they're critical. And like a preemie, when a preemie is born, part of the reason they got to put them on these little mini ventilators is they don't make surfactant yet. So they have to actually put them on ventilators because they need to use pressure to keep inflating their lungs because they can't just breathe and fight the surface tension. So I should also mention one other thing about this lung tissue. There's also kind of elastic fibers around. Those are actually important. So that means that the lung tissue is kind of stretchy, and we call it compliance. Compliance is the stretchiness, like how much it stretches out when you are kind of inflating it. And what this means is if the lung is inflated and then you just let it go, it naturally wants to collapse in because this elasticity, the natural elasticity, if you start inflating this thing and all else being equal, no other forces, it's going to want to just collapse back in. The residual surface tension, even though you've broken up most of the surface tension with surfactant, there's still some surface tension. That is also going to serve to want to make the alveoli and the lungs get smaller. So when we talk about pulmonary ventilation, this is going to be important, that the lungs on their own want to just get kind of smaller on their own. And so this brings us to COVID. Finally. So it's basically taken out surfactant cells. So you're not making surfactant. You are not breaking up the surface tension of the water, which means it's now really hard to inflate your lungs. So the same pressure differences you would normally use that would just drive normal pulmonary ventilation are no longer inflating the lungs the way they were. So that is a big part of the problem. And we're going to see there's even a worse thing about it. And we'll come back to this little evil dude in a little bit when we're finishing up talking about what's driving actually breathing, like the neural control of breathing. Because the particular way this COVID pneumonia works is different from a lot of more typical pneumonias, and people don't actually realize how sick they're getting until they're really, really sick. And we'll talk about why that's true. All right. So I think we are done with the alveoli. So now we just have to look at pulmonary ventilation and literally like 3 minutes of neural control of breathing. So let's talk about pulmonary ventilation. Again, that means just inflating, deflating the lungs. All you need is, so if we have our person here, we're going to make a highly simplified version of a person here. Chest cavity. Here's the diaphragm. So this is the thoracic cavity. We need to give him some lungs. And let's be more specific. This is the diaphragm muscle. What nerve innervates the diaphragm?
Phrenic.
Yeah, the phrenic. And when the diaphragm contracts, it actually flattens out. So it's a little non-intuitive. Normally you think of things getting smaller when they contract, but the thoracic cavity is going to enlarge when the diaphragm contracts. So when the diaphragm contracts, it tightens up, it's actually pulling down. It means the volume of the thoracic cavity is actually rising. That's going to be the main thing driving your breathing is actually the diaphragm contracting. As it pulls down, the volume of the thoracic cavity gets bigger. What do we call the serous membrane that surrounds the lungs?
Pleura.
Yeah, the pleura. So the pleura, we're going to have a parietal pleura that's against the body wall. And we're going to have a visceral pleura that's against the lung tissue itself. So there's visceral and parietal. And there's this fluid with the pleural fluid in between. And that fluid is really, they're right up against each other. So they slip and slide, but if you try to pull them apart, there's a suction. So there's a really strong suction in between the two. So what that means is as the thoracic cavity changes size, when you enlarge it, the lungs will get pulled along because of that suction between the lungs and the body wall there. So the lungs like don't have any muscles to change size or anything, but your thoracic cavity has lots of muscles to do stuff. So you can have the diaphragm go down. What are other muscles that change the size of your thoracic cavity?
Your
The
intercostal
intercostals,
muscles?
totally. Intercostals. What other ones can yank on your ribs?
Serratus anterior.
Serratus anterior, pec minor, the scalenes, all of those can all change the volume of your thoracic cavity. Actually, even just your back muscles, like you can just by leaning back and stuff, you can like kind of expand things, stand up straight, whatever. So we've kind of set the stage here. We've got the lungs here. We've got the chest cavity. We have the movement of the chest cavity coupled to the size of the lungs by this suction between the pleura. And basically, I'm just going to type it out so we can just get through this a little faster. Kind of inspiration. If you want to breathe in, the first thing that happens is you have muscles contract that are going to enlarge the volume of your thoracic cage. So the main muscle driving this is typically the diaphragm. So diaphragm and other muscle. Increasing volume of the thoracic cavity. That's part one. Two, because of that suction, the lungs are going to get pulled along and are also going to increase volume. So now the lungs have increased in volume, and I can like, this is B1. And actually for completeness, we need to talk about Boyle's law real briefly, which I'm sure most of you probably remember from, I think you've probably seen this in just Bio 110 even, because I know you learned this stuff back then as well. Boyle's law, if you have some gas, the pressure is inversely related to the volume. This is pressure and volume. And it's pretty intuitive. If you have a gas. A gas can be thought of as just like a bunch of little particles zipping around in the, through space. And pressure is just force per area. So as each of these little particles wax on the wall of whatever little box they're in, the gas is exerting a pressure. If I put the same amount of gas in a much larger box, these little dudes are going to spend much more time just traveling through space and much less time whacking it on the wall, exerting pressure. If I take them and I put them in an even smaller box, they're going to constantly be whacking on the wall, be even higher pressure. So larger volume, less pressure, smaller volume, more pressure. So that is pretty intuitive, I think. So here, diaphragm contracted, we increased volume of the thoracic cavity, the pleural suction, lungs are bigger. So 3, now that the lungs are bigger, what's going to happen to the pressure in the lungs?
Will go down.
Pressure in lungs goes down. And so now you have this low pressure zone in your lungs compared to outside. Now it's lower pressure in your lungs than the pressure outside, and the air will just rush in. So the critical thing to remember here is the kind of the causality. What causes what? You need to make sure that you don't think of the lungs like a balloon. Like a balloon, you blow air in and increase the pressure, which then makes the balloon get bigger. This is very different. This is, you are actually pulling the lung into a large volume, which creates a pressure, low pressure zone in the middle of the lung, which makes the air rush in to fill that low pressure area. So like if on your exam, there's a question about describe how pulmonary ventilation works, definitely do not say, oh, the air comes in and then the lungs get bigger. It's like, no, that's not true. It's like the lungs get bigger, increase volume, which causes low pressure, which then makes air then come in. So be really, if it's, yeah, I'm gonna, does that make sense? Or are there questions or clarifications I can make? Cool, so we call that negative pressure breathing. Some, like a frog actually does do positive pressure breathing. It'll like inflate its lungs by gulping air. Moving up its lungs like a balloon and then letting it out. But that's not us. Again, I can change the volume of my abdominal cavity by using my abdominal muscles. And as my stomach changes size, it'll create a low pressure zone in my stomach and the air will rush down my esophagus. Right? It's just basic kind of physics. When you want to exhale, it's just the opposite. Basically, you're going to relax the muscles. Your thoracic volume decreases. The lung volume decreases. And the lung volume is going to decrease because it naturally wants to go down. As you are letting the thoracic cavity get smaller, the lungs are just going to, they want to get smaller. So they're just going to keep getting smaller as much as they can, kind of limited by the suction by the current size of the thoracic cavity. As the lung volume goes up, lung pressure is going to go up, or the pressure in the lungs goes up. What am I saying? The lung volume goes down, the pressure in the lung goes up, and therefore the air is going to flow out. So that is that is that. So basically, as your thoracic cavity gets larger, the lung gets larger, makes the pressure drop, air flows in. When you relax the thoracic cavity and it gets smaller, the lungs naturally are trying to get smaller. And as they get smaller, the pressure builds up and then the air leaves. What happens if you get shot in the chest or stabbed, let's say stabbed or whatever, but there's a hole right here? Ouch.
The volume can increase.
So what do you mean by that exactly?
The
Uh,
lung collapse.
Go ahead, Elsa.
Is the lung will collapse?
It can't retain a pressure in the lung?
Right, so remember, the reason why the lungs stay the size they do is because of the suction. They naturally want to get smaller. So if you break that suction by putting a hole right there, all of a sudden air is just going to rush into the space between the two pleura and the lung will collapse in. So the pleuras, the parietal pleura and the visceral pleura are no longer have suction with each other because now we just got air that rushed in through that hole in the chest wall here. So whenever you get that happening, it's called a pneumothorax. It means air in the thorax. It leads to a collapsed lung is called atelectasis. So if this happens, luckily the pleura on the two sides are independent, so you can actually collapse one lung and still have the other side marginally working and ventilating. But as long as this suction's gone, it doesn't matter how much you move your diaphragm up and down. All you're doing is bubbling out your hole. You're not actually changing the size of your lungs, so you're not actually ventilating at all, right? So the diaphragm only has an effect on the lung size as long as the suction is there. So thoracic volume is coupled to lung volume. When this happens, they have to basically they have to patch the hole and drain all the air out and restore the vacuum in there, restore the suction. So that's like the basics of pulmonary ventilation. Are there any questions about that?
I'm just curious about with the repair of that, then would the different pleura then be repaired with serous membrane or the serous fluid in between come back to then form that vacuum on its own, or?
Yeah, so I think like what they do is they what do they call it? They call them occluding.
Right, occluding.
So and then they have like a little valve thing that can drain the air out of the space. And eventually I think the tissues just heal. You constantly have the serous fluid being pumped out. There are processes that are like little bailing pumps constantly making sure that that vacuum is maintained in there. So I think once the tissues heal, the system just starts working properly again.
Okay.
You can also have the vacuum broken if you have holes from the inside as well. You can get pneumothorax if there's some like problem with the lung tissue where the air actually gets from the inside as well. So let me see if there's anything else I want to say here. Oh, so yeah, so that's because the lung tissue naturally wants to get smaller. Exhaling is usually passive. That's what I want to write. This part here, this is typically passive. Obviously you can make it active. I was actively exhaling. But when you're just doing quiet breathing, if you just relax your muscles, then the thoracic volume will go down. The lung naturally is going to get smaller and you breathe out. People who have like emphysema where the lung loses its elasticity, then that's not so true. If you have emphysema, then you relax, and because the lung doesn't naturally have that elasticity anymore, you actually can have to be more active in your exhalation as well. So I'm going to be very kind of brief here. Basically, in the pons and the medulla, there are a variety of respiratory control centers. And the main take-home message is that your feeling you need to breathe is not about levels of oxygen. It's driven by a rising amount of CO2. So you're in general, you're not measuring oxygen, right? You can actually put an oxygen mask on somebody and they'll still keep breathing fine, even though they're breathing like pure oxygen, because, if you don't breathe, CO2 starts building up in your body, and that's the trigger that tells you that you need to breathe. So this is, I'll put a star, this is important, that CO2 drives our need to breathe, not oxygen. And then it takes us all the way back to this little puppy dog here, or evil puppy dog, the COVID. So as COVID is messing with surfactant cells, which means your breathing and your lungs do not inflate that well anymore, but it actually doesn't mess so much with the compliance, the stretchiness of your lungs. So you're breathing, and yet when you are exhaling, your chest cavity is going down and the lungs are getting smaller and pushing out the air that's in there, so you don't have a buildup of stale air. And because there's no buildup of stale air, you do not feel like you got to breathe. So if there's no building up of stale air, you don't feel you need to breathe. But what you're not noticing is that your lungs haven't been inflating very well and you're not getting enough oxygen. So there are others. So what happens is people actually don't realize that their respiratory function is seriously compromised until things are really bad. By the time they realize something's wrong with their breathing, or they feel like they're having trouble breathing, things have gotten way farther along than you would have imagined in terms of the damage that's happening in their lungs. One of the things that this is something I have, all my physiology teachers at College Marin all have these at home now. The little pulse oximeters. You put on your finger, as long as you look at there, you should have somewhere around, mine says 98, that's good. It should be around that. Some of these people, by the time they realize something's wrong, they're that 98 means 98% oxygen saturation in the blood, which should be good if your respiratory system is working well. It means you're bringing oxygen down, the oxygen's getting into your blood and keeping the oxygen levels up high to deliver to your tissues. But for the people who have the disease, you can start getting lower and lower levels of oxygen actually in their blood to get delivered. And they don't even know something's wrong other than they're just maybe feeling off. But they don't actually feel like I'm having trouble breathing or my oxygen's down because they are still ventilating and the lungs are still expelling the CO2. So that's another one of these interesting things about this COVID-19 disease that, ties into, kind of what we're learning about this respiratory system is part of the reason people get furthered into this illness before they even realize, they're showing like the majority of the people who end up on respirators, something like more than 80% of them actually die. Like by the time you're on a respirator, the chances are, kind of 4 out of 5, you're not going to make it. And, it's why it's good having a pulse oximeter so you can actually see if something's off. You can check your fever, but you might want to check your blood oxygen levels as well and make sure they look good if you're feeling off at all. So any questions?
I read in an article from New York Times that like people's oxygen levels were getting down to like 50. Is a
Yeah.
normal level still like in the is it like low 90s is a normal level?
It shouldn't even be low 90s. It should be kind of upper 90s, like mid 90s to high 90s. Yeah, no, so I read that same article. It was really scary.
Yeah, they were calling it like silent hypoxia.
Uh-huh. Yeah, no, so I think it's really worth paying attention to that. Like I said, all of my colleagues at College of Marin went back to school and went to lab and got our little pulse oximeters.
This might be going too far into it, and so you don't have to answer, but I am interested in terms of the connection with the heart too, because it seems like there's that direct correlation with it being the heart getting impacted as well with COVID.
So that I don't know.
Okay.
And I also know the virus enters the cells through, it's called the ACE2. So there's this enzyme that's bound on the membrane, mainly of lung tissue, but it's also found in lots of tissues. And the virus can attach to that enzyme, that membrane protein, and use that to enter the cell and infect it. And that particular membrane protein, there's a lot of it in the lung tissue, but it is found other places as well. So it's not surprising that the virus is causing problems in other tissues or other systems because it doesn't just hit lung cells. That's kind of the main thing that we've been following, but it's probably messing other things up as well.
I read that it can it can cause blood clotting. Could that be related to the heart?
Yeah, I don't know. I mean, you can have aberrant blood clots if the heart is like arrhythmic or something. But I don't think the heart directly in general it'd be probably more something about platelets or something like that. Yeah, I don't know. I did see that, but I didn't follow up on that yet.
I thought I heard like in regards to that it wasn't people were dying of pneumonia. It was that they were clotting in their coronary sinuses, which would be like the heart. And that's what caused them to eventually die.
Interesting. That I haven't read. That's fascinating. So, yeah, I mean, we're still figuring this thing out. But in general, definitely the thing about dealing with surfactant cells and making it hard to inflate your lungs is a big part of it. And yeah, it seems like there's lots of other things that are going on with it as well that we're still learning.
Just to clarify, when you say stale air, that means like a high content of CO2 in there, and so then you wouldn't know that you have that, and so you wouldn't feel the need to breathe.
Wait, so say that one more time.
Can you just clarify when you said stale air, and you said that there's no buildup of stale air, like what you mean by?
So like, let's say these are your lungs. You know, if you stop breathing, the CO2 starts building up more and more. Your blood is letting go of CO2. So I consider stale air is the air that is loaded up with CO2 that you need to get rid of. So then you kind of blow that out. And you don't actually blow all of it out. You blow most of it. Obviously you can't because there's dead anatomical space here. You can't empty everything. Then you bring in the fresh air that mixes with a little bit of what I call the stale air. So it's mostly fresh. And then carbon dioxide still starts getting loaded into that, but then you blow that out and bring fresh air. So you never have this buildup of what I'm calling stale air, of higher concentration of carbon dioxide levels that just stay. Because every time the carbon dioxide levels start building up, you blow out the stale air and breathe in the fresh air. That's how I was using it. If does that clarify?
Yeah, that makes sense. And so then can you explain then with COVID that
Right.
I thought you said there's no buildup of stale
In
air.
COVID, a lot of times in pneumonia, the lung tissue is just kind of losing its elasticity. It's making it harder to inhale or exhale. And part of what happens is you actually get a buildup of carbon dioxide because you're having trouble with that exhaling, kind of getting rid of the stale air. But with the COVID, at least in the beginning, it doesn't affect the elasticity of the lung tissue that much. So the exhaling works really well. So even though the inhaling, you're not actually getting so much oxygen in as you normally would have got, you're still exhaling efficiently. You're not building up excess carbon dioxide in your system. So you don't feel like anything's wrong, even though your oxygen that is coming in is less than it should be. Does that make sense?
Yeah, that makes sense. Thank you.
Sure.
As a point of reference, I was sick back in December, and in February I went to see my respiratory doctor, and my oxygen saturation was like 92, 93%. And I had been like really fatigued. It was really easy to get fatigued.
Right. So it's pretty, I mean, there's more and more we're finding. It's been around. I mean, I know some people who, I have a friend who's a teacher who said that she was out of breath trying to teach her class back in like early February. Like couldn't, not only could barely get upstairs, but actually just trying to lecture was running. So I think is probably, I mean, once they have the antibody tests and we'll see who actually has had it, it wouldn't shock me if a lot of people have already had this already. Yeah, it's going to be, hopefully they get these tests online and available soon. Just also so we can get out of this shelter in place stuff as well. So, cool. Any other comments, questions about this stuff? Okay, so for labs, let's see what the time. Should we take another little break or just go into the lab stuff right now?
I say just go into it.
Okay. All right, so I set up a few different things in this week's module that will be useful. Let me get them going. Okay, hold on. I need to log back in. Zoom. Okay. All right, modules. And so we'll do the cadaver ones last. I'm going to just go to virtual histology lab. I mentioned this before. They actually have nice pictures for the respiratory system, or nice slides. And again, the thing I like about this virtual histology lab is it actually feels more like actually using a real slide instead of looking at a picture of a slide. Because on a real slide, you zoom in, you zoom out, you see the big picture with the context, you go in and you see the details of the cells in the epithelium. So looking at a picture of a photomicrograph is just so, it's just so unsatisfying compared to actually looking at real slides. And this at least gets you a little closer into kind of the fun part of looking at microscope slides. So if you go into the slide box, we can go into respiratory system here. We could look at the epiglottis if you want. And again, what kind of tissue should we have making the kind of foundational structural core of the epiglottis?
Connective tissue.
No. I mentioned
Smooth muscle.
no. All of the so it's
Cuboidal.
so hold on. What kind of tissue makes up most of the kind of the skeleton of the larynx? The thyroid blank, the cricoid blank.
Cartilage.
So it's cartilage. It's all cartilage, but what kind of cartilage makes up the core of the epiglottis?
Elastic cartilage.
Elastic cartilage. So as we kind of zoom in here, it's like, oh my God, I remember this stuff. Remember that? It's got the lacunae and the little fibers. So there's elastic cartilage in there. This should be ringing some bells. If we go and look at what kind of tissue actually makes up the covering of the epiglottis?
Stratified squamous.
Totally, stratified squamous, right? It's because it's coating your oral cavities, the back of your throat there. So this is really nice. You can see the stratified squamous covering it. You can see the elastic cartilage forming its kind of core. So you got that going for you. Trachea. When we look at the trachea, we're going to find
Hyaline cartilage.
here's hyaline cartilage. So it's smooth. There's no little fibers. And then when we go to the epithelium, it's like, oh my God, there's pseudostratified columnar epithelium. And you see the little cilia. So this is exactly what we would have expected. In the lung, it looks like a bunch of little rooms. These are what the little alveolar sacs look like. So basically, you've got the little alveolar ducts, kind of the hallways, and then the alveoli themselves are the little rooms, the little bubbles. As you move around, this would be a bronchus here. This is one of these larger ones where we still have the respiratory epithelium lining the middle of it, or the lumen of it. As we move around, we can also find this thing here. And what is this thing probably?
Simple squamous epithelium.
So actually the lining is going to be simple squamous, and this is all muscular. This is a big blood vessel. This is probably one of the arteries. Because again, if you look, it just looks like smooth muscle in its wall. Here's another. This is probably a vein. It's got smaller walls, thinner walls, I should say, filled with blood cells. And it does not have cuboidal or pseudostratified columnar epithelium lining. This is a blood vessel here.
Wait, I thought arteries are circle and keeps it shape.
It depends. You know, in general, that's true. But if as you're making the slide and squishing something, it can get squished.
Okay.
This is a vein. This one here, I would say, is a vein. I'd say this is probably an artery, though, given how thick the walls are. It's just a squished artery. So it's kind of fun. And as you walk around, as you run around, you can find smaller, like here, this is actually a much smaller blood vessel. You can see this is like a little arteriole probably with some blood cells in there. When you get even smaller, you can probably find, I mean, these are probably capillaries with little blood cells trapped in there. So it's fun to zip around in here and check things out. We can look at another lung. Again, you can see little blood cells. These are probably in little capillaries that are just running around the little boundaries in between the alveoli. And then some blood cells that have just spilled out. Here's definitely a little arteriole or a little venule, I'm not sure which. A little bit of smooth muscle making the wall and some blood cells stuck inside. Here's a beautiful little venule. You can totally see a little bit of around the wall and then the blood cells stuck inside. Right, so spend some time moving around. You can definitely see the alveoli. You can see blood vessels. You can also see in a lot of them the bronchioles. Like if we go over here, this is now a bronchiole because it's got this more cuboidal epithelium lining it rather than just endothelium with smooth muscle in the wall. So if we look at a tube like this, where you've got this more cuboidal epithelium, then we know this is like a little bronchiole, an airway coming into the lung tissue. Whereas, you know, over here, smooth muscle with endothelium, simple squamous lining with some blood cells. This is our little artery or arteriole if I've ever seen one. Right, so have fun, kind of zip. Oh, here's again, a little venule with just some endothelium and some blood cells in there. This is probably a smaller little bronchiole. You can still see some little ciliated epithelium. So this is one of the larger ones before it's become the really dinky little bronchioles. So play around, see if you can recognize the tubes, get a sense for the big picture. You know, I would definitely feel fine putting a slide like this on your test and asking, what kind of tissue is this? It's very distinctive. So definitely you should know lung tissue. What else do we have here? So that's the rest. The lymphoid tissue is actually pretty good on this one. I was actually looking at it, and when because you should recognize this is a lymph node. Remember we talked about all those lymphatic nodules. You know, this is a great picture of them with their little germinal centers. You can see the little trabeculae, the little connective tissue from the capsule coming in to kind of partition off the different sections. So these little lymph nodes are beautiful. Tonsils. How do we know this is a tonsil and not a lymph node? Who can tell me?
Is there reticular fibers in there in this space?
No, they would both have reticular fibers. Reticular fibers make up the stroma for this lymphoid tissue.
Because of the stratified squamous.
Exactly. This has stratified squamous. This is basically the lining of inside your throat. The stratified squamous of this is probably like a palatine tonsil. So it's stratified squamous with the lymphoid tissue right underneath that. That's what the tonsils are. If you go to a lymph node, Lymph nodes are little organs that are just encapsulated with connective tissue. So a lymph node has a very, lymph node is going to have just a connective tissue capsule, whereas a tonsil will have an epithelium because it's the lymphoid tissue underneath the skin. Yeah, and like I said, you are not responsible for recognizing spleen, but you should be able to recognize like a lymph node or a tonsil, I would say. Thyroid gland, this is the one that was on your test. As you get deeper and deeper in, this is a pretty stinky picture of a thyroid gland. Use the picture I gave you, but you basically have the little follicles. I don't like their thyroid gland. How about the pancreas? This is kind of what it often looks like. What are these kind of light areas that we see kind of scattered around in here?
The islets of Langerhans?
Those are the islets of Langerhans. So all these other cells are the acinar cells, the one that make the digestive juices. And those little areas they're kind of hard to see, but you still can see them. These are the islets of Langerhans. The cells also have just kind of a different look and they stain a little differently. And again, somebody who has type 1 diabetes, these are the cells that are getting attacked by their immune system. That's why they no longer make insulin. So you should look at the histology for respiratory system after I'm done talking. I need to get back to this. I want to clear that. The other thing that I kind of figured out, I spent some time this morning finding some nice pictures of things in the dissection videos. So I made where is it? Cadaver dissection videos for respiratory system. So this first one, it lasts for a little while, but it's worth watching. It's basically going to kind of show how the lungs fit into the chest and how the pleura surrounds the lungs and lines the inside of the rib cage.
We're going to move deeper into the body and expose the organs that live in the thoracic cavity. And just as a reminder of the things that you've looked at already, of course, we've got our lateral abdominal wall musculature and the musculature of the chest, including the pectoralis major muscles. You've already cut their origins and reflected those away, as well as the deeper pectoralis minor muscles, preserving their nerves and vessels. Now we're ready to remove the anterior and lateral portions of the rib cage, the bony rib cage, so that you can expose the heart and lungs within the thoracic cavity. Now we've already made the cuts, but what you're going to do to make this happen is to cut through the ribs and the intercostal muscles around the perimeter of the rib cage on each side. And if you make the cuts or your ultimate hole as large as possible by following the margin of the rib cage and then going down as far laterally as you can along the side, essentially along the mid-axillary line, you'll have nice exposure of the thoracic organs for your further dissection. Now, as you do this, if you can, you should try and get your fingers within the hole and peel the layer of tissue, the parietal pleura, away from its attachment to the inside of the rib cage so that you can leave as much of that intact as possible as you remove the bones. Now let's go ahead and take away the ribs and the intercostal muscles on each side. And you'll get an idea of what you're going to see once you've completed that part of the dissection. Now, we do have some holes within the parietal pleura here, but you can see essentially what it's going to look like. There are some areas that are thinner, almost transparent, and some areas that are pathological in this individual where they've thickened up, probably because of some sort of infection within this sac surrounding the lung.
Right, but yeah, typically it's just that super thin. It's literally simple squamous epithelium on some areolar connective tissue. It's just a super thin layer. This is the layer that is against the wall, the rib cage, the inside of the rib cage. And then there's going to be the visceral pleura that's on the lung tissue itself. And then typically there's just that suction between what she's holding and the layer that's on the lung itself.
This would be the parietal pleura, the outer layer of the sac that surrounds and protects the lungs. If we take that away, you can see the nice, delicate visceral pleura that's immediately on the surface of the lung, and we'll have a nice friction-free movement as the lung fills with air and then releases it during exhalation. The next step in the process will be to remove the sternum from the midline. And to do this, depending on whether or not the cadaver that you're working on has had osteoporosis, you may need to use a saw to cut at the inferior and superior ends of the sternum. Alternatively, you might be able to use a chisel and a hammer to cut through these areas. Again, be sure that you try and bluntly separate the soft tissue from the deep surface of the sternum as you do so. In particular, try to keep the vessels that you isolated in the previous dissection intact, the internal thoracic or internal mammary arteries, which not only supply the chest wall, but also continue down onto the abdominal wall to provide collateral circulation there. Now with the bony part of the rib cage removed, we can see very nicely the two lungs on either side within the chest and the heart right along the midline. And we have preserved the internal thoracic artery and vein on either side in this situation. So during the next step of the dissection, what we're going to do is remove the lungs. And you should be careful to, again, preserve the internal thoracic arteries. And another structure that we hope that you can keep for the entire semester if possible, and that is a nerve that you'll find traveling inferiorly to reach the diaphragm, which it innervates. And this nerve, called the phrenic nerve, travels between the lung and the heart. Okay, you're going to find one on the right and one on the left. So that as you progress to removing the lung and cutting it out of the body, make sure you know where the phrenic nerve is at all times and keep it out of the way so that we can keep that intact.
Right, so that's actually, it's very easy to see. It just runs right along the side of the heart on either side. On the cadaver, it's really easy to see. If we were doing our little piggies, it's just, this looks like a white thread running down the side of the pericardium and then just landing right on the diaphragm. Again, obviously coming from the cervical plexus.
Now to remove the lung, if you can just push it laterally, pull the heart in medially, now you can see the region of the lung called the hilus here. And this is where a variety of structures enter or exit the organ, including the airway, the blood vessels, the nerves, the lymphatics. So that what we want to do is make a nice clean cut at the hilus of the lung so we can then remove it from the body. Now you can do this in one of two ways. You can use a scalpel, or potentially you can use a very large knife that we've got, and the instructors can come around and help you with this. But again, push the lung out laterally, pull the heart in medially, and use a knife or a scalpel to cut through the structures at the hilus. Now,
It's kind of like samurai dissection. It's kind of fun.
Now removed from the body, you can distinguish the right from the left lung quite easily because they have different numbers of lobes. This is the right lung, which has got 3 lobes, and those lobes are divided by fissures. This is the oblique fissure, and this is the horizontal fissure, so that the right...
One thing you notice, I mean, the one thing that you can see just from her barely touching it with the probe, and that normally if you just touched it, you'd know it's really spongy. It's really soft and spongy. It really is mainly air spaces. So the lung tissue is super soft and springy and spongy. And so hopefully that's coming across just watching her kind of barely touch it, you could just see it wiggle and quiver underneath just a light touch.
Lung is subdivided into the inferior, middle, and superior lobes. What you're going to notice as you get the lungs out is that it tends to be darker looking on one side of the lung, and that is the posterior side of the lung, which has got more blood within it just by gravity because the cadaver has been laying on its back for some time. If we turn the lung to look at the hilus and study some of the structures that enter there, there are several structures that we'd like you to be able to identify. The pulmonary arteries, the pulmonary veins, and the bronchus, as well as lymph nodes that you find at the hilus, and in addition, a second blood supply to the lung, the bronchial circulation. So first of all, you can use several features to distinguish these different structures. The thickest walled structure that you'll find is the bronchus. Being the airway, it's got to remain patent at all times, so it's got a cartilaginous wall, which makes it very thick. So this would be the bronchus entering the lung. In intermediate thickness is the pulmonary artery supplying deoxygenated blood to the lung. And then the thinnest wall, large structures that you'll find would be the large pulmonary veins. And there should be 2 of these at the hilus of each lung. Depending where you cut through these structures, you may see more than what we just described, depending on whether these vessels are starting to branch. So look at the thickness of the walls, and also you can look at the positions of these structures relative to each other to distinguish what's what. Now, there's a simple mnemonic that you can use to determine which vessel is which, and it's RALS, RALS. On the right lung, the pulmonary artery is anterior to the bronchus, and that's what we're seeing here. Here is the primary bronchus entering the lung, and the pulmonary artery is just anterior to it. Generally, the pulmonary veins are the more inferior structures at the hilus. This is the left lung that's been removed from the cadaver, and as you can see, it doesn't have 3, but it's only got 2 lobes, which are separated from each other by an oblique fissure. We've simply got the inferior lobe and the superior lobe.
Also, I mean, you don't need to recognize, you don't need to learn RALs or anything, but it's nice to be able to see those 3 main things at the hilus there. Also, and then notice that really shiny, slippery coating. You can really notice the visceral pleura on this lung as she's kind of just moving it, and you can watch the light reflects, and you can tell it's just really slick.
In addition, you can find this little appendage dangling from the left lung called the lingula. Now let's turn to the hilus of the lung and take a look at the structures that you can identify there. Again, we want you to look for the bronchus, pulmonary artery, pulmonary vein, and some other structures which we'll look at. It's entering the artery. You're going to generally see...
I'm going to move a little faster.
Of course, it's the visceral layer of the pleural sac that's immediately on the surface of the organ. But in terms of that outer layer, the parietal layer, we can give it different names depending...
I'm not going to... You don't need to know the different... She goes, there's like a diaphragmatic pleura, but it doesn't matter. A mediastinal one. Let's keep going.
Called the pericardial sac that will reduce the friction as the heart enlarges.
Yeah, so for the rest of this, she's going to start doing the heart. So here's like the mediastinum with the heart and the pericardium. So I think it's really nice to kind of give you a sense of how the lungs are situated in there, you know, how they're different from each other, and also really getting a little more of a sense of what the pleura really is, that slippery, thin layer, one layer on the lung itself, one layer coating the inside of the rib cage with a suction in between. So as the muscles of the thoracic cavity enlarge the thoracic cavity, the lung gets pulled along by that suction between those 2 thin layers.
And contracts again.
Okay, then so this next one is really trippy. So if you have more time, you can actually just watch more of this. It's a little disturbing. It's just basically taking this head and like with all sorts of tools, and it's very, very trippy. But in the end, what they're basically doing is they're getting it so you are looking at this guy, the back of his throat, the back of his pharynx, with the rest of his body gone. So we're gonna, I'll show you what I mean. Okay.
To incise the accessory, spinal accessory nerve.
All right, so to give you a sense of what you're looking at, imagine you're looking at this guy from behind. Here's like his carotid artery. So this is his neck. And this is his throat. His mouth would be up here someplace. You know, his esophagus and stuff is coming down. So this is the back of his throat, but this guy is going to use a scalpel and going to cut into his throat from the back.
And nerve number 11 that descends obliquely, pierces the muscle, and then is obviously, it travels in that midline raphe of the pharynx to expose the inside of the pharynx. To examine the internal anatomy of the pharynx, we will need to make a scalpel incision down the midline of the back of the pharynx. There's actually a line of insertion of the right and left pairs of constrictor muscles called the pharyngeal raphe. You probably won't see that very clearly, but that's basically the target for where this incision will be. Let me go right down the midline here. And then fold back. You completed your incision, fold back the layers of the pharyngeal wall, or the constrictor muscles. And depending upon the cause of death or timing of death, you may see some debris, as in this specimen, within the pharynx and openings communicating anteriorly. I'm going to clean that up for a second and then come back and go through identifying the anatomy of this region.
Right, so it means the guy was probably like spitting up and stuff and got all sorts of. So just to stay oriented here though, again, here's the back of his ears. This is kind of the inside of his skull. His brain would have been sitting right up here. This is going up into his mouth. This little thing here is going to actually be the epiglottis. This is going to be actually his tongue coming up into his mouth. So just you'll stay oriented.
Now we have the pharynx open posteriorly and have cleaned this out. All of this area right in here where I'm laying the probe is portions of the pharynx. And the openings of the pharynx are anteriorly. One opening anteriorly is up towards the base of the skull, so I'm putting the forceps into the opening into the nasal cavity. If you look into the nasal cavity, you can actually see the nasal septum way back in there. Just the floor of the nasal cavity and the roof of the oral cavity would be comprised of what we see here is the soft palate. I'm moving back and forth. And of course, going in this direction is going into the oral cavity.
Right, so that little nubbin is the uvula, right? Is that making sense? This is like kind of, yeah.
This is literally the dorsum of the tongue that we see in this vicinity right here. You can make out the circumvallate papillae when you do this, sort of a V-shaped or chevron-shaped series of taste buds that will help you differentiate anterior 2/3 of the tongue from the posterior 1/3 of the tongue.
Right, remember we talked about the circumvallate papilla are those big honking knobs on the very back of your tongue. They're very hard to see when you stare from the front and look down and say, ah, but they're really easy if you cut the back of someone's head off.
Okay. So this is all oral pharynx, part of the pharynx that communicates with the oral cavity. This region is all nasopharynx that communicates with the nasal cavity. Inferior most component of the pharynx is down here I'm tracing as the laryngopharynx, and it communicates inferiorly into the esophagus. The opening in the laryngopharynx that also communicates in an anterior direction is right here where the forceps are, and this is the inlet to the larynx. So this structure right here, which is movable, is the epiglottis. Descending down into this.
So where does he have his forceps now? Where are the forceps going into?
The trachea.
Exactly. He's sticking them down into the trachea.
This opening is going into the airway system, the trachea, and the lungs. So laryngopharynx, oropharynx, nasopharynx. I mentioned the soft palate up here with the as a border between the nasopharynx and the oropharynx. I didn't mention the uvula, which is the little pointed piece of the soft palate. Within the oropharynx, and if we bend down the epiglottis, and I hold the head up maybe a little bit better, you can see into some depressions that are.
So yeah, we don't care about that. So that just gives you a sense. We would have done something similar like this on our fetal pigs, where you can really start to get an understanding of what the epiglottis really looks like and how it flaps over and how all the tubes are all connected like this. And there's just one more of these videos that I think is worth seeing here. And this one is going into the nasal cavity, really showing the conchi well, and also kind of giving you a sense of where those little, I said those little passages that connect to all of your sinuses, and the ones that get clogged up when you get a sinus headache and can't equalize the pressure in your sinuses.
And you recall that the skull has been sawn in half, so I'm going to flip this down to expose the two medial surfaces of the skull. Looking inside, there are some familiar structures from the last dissection. You'll recall the levator palatini muscle and the tensor veli palatini muscle, both of which are originating from the lateral and inferior aspects of the auditory tube. And here's a familiar structure, the opening to the auditory tube.
Okay, so he's sticking his little probe into the entrance to the eustachian tube there.
Now, this first part of the dissection is to inspect the nasal cavity and look at some of the structures that are in the nasal cavity and try and get a handle on the structures that drain into the nasal cavity. First of all, there are three spaces created in the nasal cavity by these protrusions into the nasal cavity known as the concha. This is the inferior concha, this is the middle concha, and this is the superior concha. And the spaces underneath each of these concha are known as the meatuses, this then being the inferior meatus. If I flip the inferior concha up, we can look into the inferior meatus. One structure drains into the inferior meatus. That is the nasolacrimal duct, which comes from the lacrimal sac that you found when you dissected the orbit. So that would be the only structure that drains into the inferior meatus of the nasal cavity. The middle meatus of the nasal cavity is a little bit more complicated, and if we elevate the middle concha, we can look underneath of it and we see that there are actually a number of openings into the middle meatus. The middle meatus drains the frontal sinus into this groove known as the hiatus semilunaris. This is the groove of the hiatus semilunaris. And the frontal sinus drains directly into that groove. It also drains the largest sinus of the skull, the maxillary sinus. The maxillary sinus opens directly into this area underneath the area of the hiatus semilunaris as well. And then the anterior and middle ethmoid cells open onto this bump, which is known as the bulla ethmoidalis. Those are all the structures that empty into the middle meatus. You will also possibly be able to see the superior concha and the superior meatus in some of the cadavers, but in others it gets difficult to see. We can get an idea of what it looks like in this particular cadaver.
So just to kind of give you a sense, the conchi, or concha is what he's calling them, this is where those sinuses, those hollow spaces in your skull bones actually communicate with to equalize pressure. It's where that nasolacrimal sac drains into. Oh, what's the official word for nose hairs?
The vibrissae.
Exactly, the vibrissae. So anyway, so I just kind of, just to give you a sense of what this looks like from this side. So what I would say at this point is you should probably just spend some time trying to get a sense of what lung tissue looks like. Make sure you understand what the trachea looks like. You should understand the basic kind of anatomy of the pharynx, the nasal cavity to the pharynx to the larynx. And yeah, and in terms of stuff for me to go over with you, I think that's about it. But I think between the slides and the cadaver things and then just your books and things like that, you've got plenty to just kind of dig into with all this. The last thing, are there any questions about all this before we go over the lab practical? Okay. So lab practical. I did not finish grading the lab in the lecture exam yet, but I actually did finish grading the lab practical. I went through and, double checked, made sure again, if someone wrote red blood cells instead of red blood cell, it marked it wrong. So I was, I made sure I went and corrected, made sure everything was the way it should be. So I'm going to stop the recording for this part. Where's my